This is our mitosis versus meiosis slideshow by Jenna and Kiara.
The cell cycle. The longest phase of the cell cycle is interphase. Inside interphase is the gap 1 phase, where the cell grows. Then there's the synthesis phase, where the DNA replicates. And then the gap 2 phase, in which the cell prepares for division and the organelles replicate. Then there's cell division, and inside cell division there's the M phase, in which the cell divides.
Mitosis includes asexual reproduction. The advantages include fast reproduction. The disadvantages include lack of genetic diversity.
Phase 1, prophase. In this phase, the genetic material condenses and the duplicated chromosomes become visible. Phase 2, metaphase. The centromeres of the duplicated chromosomes line up across the center of the cell.
Phase 3. In this phase, the chromosomes separate and move along the spindle fibers to opposite ends of the cell. Phase 4. Telophase. In this phase, the chromosomes begin to spread out into a tangle of chromatin.
Cytokinesis. In cytokinesis, the daughter cell splits apart after mitosis. In animal cells, the membrane is drawn inwards until the cytoplasm is pinched into 2 parts. In the plant cell, a cell plate forms halfway through the nuclei, making it develop into the cell membrane and forming 2 new cells.
Meiosis performs sexual reproduction. The advantages of this include genetic diversity. However, the disadvantage include more time required to reproduce.
Phase 1, prophase. In this phase, the replicated chromosome pairs with its corresponding homologous chromosome. In this phase, crossing over occurs. Crossing over is when pieces of the homologous chromosomes are exchanged.
Phase 2, metaphase 1. In metaphase 1, one pair of homologous chromosomes line up across the center of the cell.
Phase 3, anaphase 1. In this phase, the spindle fibers pull the homologous chromosomes apart and towards the opposite ends of the cell.
Phase 4, telophase 1. In telophase 1, the nuclear membrane forms around clusters of chromosomes. In cytokinesis, 2 new cells are formed.
Phase 5, prophase 2. In this phase, the chromosomes, which are made of 2 chromatid, become visible.
Phase 6, metaphase 2. In this phase, the chromosomes line up in a single file line across the center of the cell.
Phase 7, anaphase 2. In this phase, the paired chromatids separate to the opposite sides of the cell.
Phase 8, telophase 2. In this phase, 4 daughter cells are produced. These cells develop into gametes. Gametes in males is sperm. In females, they are eggs.
Mitosis versus meiosis.
Mitosis is used in asexual reproduction, growth, development, and injuries. It has 1 division and 2 daughter cells that are diploids. It has 1 parent that splits into 2 daughters, containing 46 chromosomes.
Meiosis is used in sexual reproduction and results in genetic information. It has 2 divisions as well as 4 daughter cells, which are haploids. The parents in meiosis contain 46 chromosomes and the daughters contain 23.